Sir, you may continue now.
Okay, thank you so much. Yeah. So yeah, yeah. So okay, yeah. So I'll be good afternoon, everyone. So I'll be talking about genetically encodable fluorescent RNA sensors. So we all know nucleic acids, they are kind of like a beauty at nanoscale, right? And because this conference is about the nanoparticles, and maybe I can say that the DNA, if I talk about that deoxyribonucleic acid, the DNA itself, I usually consider, because I work on the nucleic acids, I consider DNA also as a kind of a nanoparticle. Because if you look at the dimension of the DNA, it has 2 nanometer width and has 3.4 nanometer, say, one turn of the helix, or you can say its bases. We know that A, T, G, C base pair, they are 0.34 nanometer apart. And the good thing is this length, so 3.4 nanometer, you put like 10 bases, it will be 3.4, you put 20, it will become double of that, you put 30. So it's a dimensions you can easily change. Of course, not the width, but length. And then you can draw also interesting structures from the DNA, like the DNA nanostructure, which I'll be showing you in the next slides. Okay. And the people agree that DNA really looks beautiful. I'll show you some examples. How can, if I'm saying that. Okay, so there's a helix bridge in the Singapore, which is looks like a DNA shape. There's a helix pedestrian bridge in the Seattle, right? There is a double helix bridge in China. There is the Evolution Tower in Moscow. There's the Agora Garden, Taipei, which is basically inspired from the DNA double helix. And even at ISA Tirupati, because I'm from ISA Tirupati, we are also planning our library. It's a model of our library. Our new campus is getting ready. So we are also planning kind of a double helix stairs for our library. So this is ISA Tirupati library, how it would look like. Okay, so we can see that the people are always, they like the beauty of the DNA. And the another property in the DNA is its base pairing. It's a very, very good thing that how A can bind to T and G can bind to C. I'm sure like all of you know, might be thinking A binds to T and G binds to C. That's how the DNA structure. You have thymine binds to adenine, guanine binds to cytosine, right? And same way, if I have a strands of helix and I leave a sticky end, we call them hybridization using sticky ends. If I leave a sticky end, it's like a double helix, and then you have a single helix here, having GTCAT sequence, for example. You can have another double helix, which is extension over here, which is complementary to this. And they will, of course, they both will hybridize. And that's we say we leave a sticky end here so that the other sticky end can bind to it. They'll, like the glue, they will stick to each other. And this is the property in the DNA, because the A recognize T and the G recognize C, we can use in the nanotechnology to build different structures. Like in see here, you can have 4 strands, you can have sticky ends, they will combine to each other, make kind of a lattice, which you can see in the microscope using AFM, atomic force microscopy, cryo-EM, or the other instruments. You can see a nice structure coming up because it will keep on becoming more and more joined, one joined with the another, with the other. So that's how you get the structures, right? Even we can give any kind of shape to the DNA because it's a DNA single strands. And we know that A binds to T, G binds to C. That's all the knowledge we need, basically, right, to work with. So we can easily plan the sequences on a paper, and then we heat the sequences together. They will bind, forming triangle, square, 3-dimensional structures like tetrahedron. All these are nanoparticles of defined size. We can tune their size, we can tune their properties depending on the need. Right? These are other examples that people have made different kinds of nanostructures out of DNA. Right? Similar way, now I'm coming to the second point, because we know the central dogma where the DNA gives RNA, right? Can we do the similar things with the RNA? That's a question. Of course, yes, we can do, right? Because the RNA also, instead of T, there's a U in there. So A binds to U, G binds to C. The things goes exactly the same way. And that's how you see that A binds to U, and we can have various different kind of structures, even from the RNA too, right? And RNA is not like a single strand like we usually see in the textbooks. It's not like that. It has like a structures. You will have multi-branch junctions, you have bulge loops, you have pseudoknots, you will have hairpin loops, lots of different structures in one piece of RNA, right? And what you can do is there's an interesting thing in RNA is called kissing loops. If I have one RNA and the second RNA, it looks like these two RNAs, they're kissing each very, very stable complexes, because if you look at the structure of this, it looks like a single double helix, like a connected together double helix. but it's not connected together. You can see green is separate, this portion cyan is separate, like here, and they are just by hydrogen bonding. And because of that, it becomes very stable. And if you use the property of this kissing complexes RNA, you can have one RNA, second RNA, third RNA, where they are kissing at this position, and this position. You can generate a trimer. You can generate a pentamer. And that's how in the microscope, you can see the pentamer coming up, a heptamer coming up, tetramer, hexamer, foot-to-foot trimer, any kind of, the symmetry you can design like on a paper. So, and we can make with the RNA sequence the similar thing, and we can see in the AFM, the similar things are coming up, right? And now I'll take you one more little forward to the another structure of the RNA, which is called the G quadruplexes structures. So, the DNA exists as a double helix, right? But DNA or RNA, the 4 strands of DNA can also come together, or 4 strands of RNA can come together to form a G quadruplex structure. It's basically, you see, 4 G's are coming, G, 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 by Hooke's-Steen hydrogen bonding. There's a special kind of bonding here to form this kind of a G quartet structure. And this is one stack of G quartet, second stack of G quartet, third stack of G quartet. That's what we call quadruplex. So G quartet, which gives us a G quadruplex. And these are different topologies, this G quadruplex. It has a parallel topology, different kind of, formations, parallel, anti-parallel, depending on how the strand is going, all the 4 strands. You can see here the first strand, second strand, third strand, 4, or it can be a single strand also going 4 times, right? So this is a G quadruplex, right? So we understand DNA, we understand RNA, we understand G quadruplexes now. And this G quadruplexes is there in an RNA. We name the RNA baby spinach. Very funny name, right? So it's a baby spinach RNA, which has that G quadruplex in there. Now I'll tell you what is this baby spinach now. So this baby spinach RNA, it's basically a RNA. We call it spinach RNA. So this spinach RNA, when it binds to a small molecule, this is DFHBI, this molecule is not fluorescent. You all know fluorescence. So this molecule is not fluorescent, but when this RNA binds to the molecule becomes fluorescent. It starts showing fluorescent. For example, you can see this is my molecule, this DFHBI molecule, not fluorescent. This is my RNA, not fluorescent. You mix these two together, like here I mixed both the things, becomes fluorescent. If I use any other RNA, which I call control RNA, any other RNA instead of spinach RNA, again, no fluorescence. Only this RNA, this fluorophore will give fluorescence. So that's why because this gives a green color, the scientists, they named it like, because it looks green color, like a spinach we eat, right? The vegetable. So they called it spinach aptamer. That's nothing to do with the spinach vegetable, just the color, because it's a green color. It's easy to remember what color spinach gives. I can say green, right? The people try to make this RNA a little bit smaller, and then they call it baby spinach. And as I told you earlier, the baby spinach has a G quadruplex structure, right? The other way the people selected other RNAs, we can call it broccoli. Broccoli means it will be a little bit more greener. Now, if I ask you, I have a mango aptamer, which is there in the literature, right? So you can understand what color fluorescence that mango aptamer will be showing. Yellow fluorescence, right? Because mangoes are, so pepper, there's a pepper aptamer, there's a corn aptamer. It's easy for us to remember without even looking at the sequence. That's why people, they like to give the funny names so that it's easy for remember us. So broccoli, chili, many different kinds of aptamers are there. Okay, basically RNA which bind to a fluorophore and then lights up. We also call them light up aptamer, because nothing is fluorescence. Once the system binds, it shows fluorescence. Now, our thing is, in our lab, we are trying is, these G quadruplexes are very, very important structures. Okay, they have at the ends of the telomere in the DNA, right? And even in the gene promoter, they regulate the gene expression. Many, many things these G quadruplexes do. And people are trying to see how we can stabilize these G quadruplexes, right? In our lab, we are trying exactly opposite. Almost the whole world is doing, I'll say. If you see the G quadruplex, you'll get hundreds of paper, G quadruplex stabilizing ligands, but you'll not find disturbing. If we can change the gene expression by stabilizing it, we can also change the gene expression by disrupting it, right? That's a simple idea. So we thought how we can disrupt the G quadruplex. Now, the easy ways to disrupt the G quadruplexes. And what we do is, simple thing is, it's RNA. If I put an extra sequence here, make it a little bit longer on one end without duplex, it can disturb, because every RNA will have a different secondary structure. So we thought we took this RNA, we put this extra sequence to it. And now because this extra sequence is attached to it, this DFHBI will don't like to bind it. It says you are not the original one. You have some extra sequence to you. I will not bind with you. Okay, DFHBI will not bind to it. And what we get is no fluorescence there. But as soon as you have something, this red strand, which is complementary to green strand, now because red is complementary to green strand, and that will make a duplex. So basically this duplex will get extended, right? So this duplex comes over here, and this takes up the same shape like earlier one, and then this can bind to it because the pocket gets regenerated, it binds to it, and we see fluorescence. So very, very simple thing. This RNA does not have any modifications in it. Pure normal RNA, regular RNA without any modifications. So with just a simple RNA, I can detect a DNA or RNA. This RNA can be a COVID RNA if you want to detect COVID, right? Or this RNA can be a microRNA if you want to do the early detection of diseases. And we tried with DNA, RNA, all the combinations, and that's how it is. We reached the so this you can see here. It gives fluorescence. All the data is showing the same thing. There is no fluorescence in the beginning, and then once you add with the DNA or the RNA, you see the fluorescence increases a lot. Then here, okay, almost I think 28 times the fluorescence increases. And the same thing we showed by gel that it works. And the limit of detection, how much RNA we can detect, it's basically 2.5 nanomolar we can detect, right? There's a mutation analysis. If some mutation is there, it will not work. With the two mutation, it doesn't work. One mutation, it works less. Okay, so that's the thing. And also because it's a nascent RNA, you can even express this RNA directly into the cells using a plasmid, right? So that's also because the miRNA detection is a very difficult task, right? It's a very small RNA. It's very difficult to detect. So basically, what we're doing is we are disrupting the G quadruplex by adding a sequence so that this target, this green thing, which is here, you can see it does not bind to quadruplex, and then making it regain the structure by attaching a DNA or RNA. That's how we're detecting DNA or RNA. So that's a one part of the work. We tried many different RNA. So you can look at the paper also, and there's some unpublished data also, which we'll be publishing soon in next few months. Okay, and another, so this is one way, one project where we used to do that. A similar one last, I think 2 or 3 more slides. So another way we thought like, okay, that's the one way we can disrupt the RNA. What's the other way we can disrupt? Is there any small molecule, some ions which can disrupt? And we found that that simple silver ions. So they bind to G guanine base like this. You can see guanine bases bind to silver. So because they bind to silver, we thought that if we add silver in there, it should disturb the G quadruplex, right? And once the G quadruplex is disturbed, that molecule will not bind here. It will open up and it will not show fluorescence, right? So when we added silver to it, the same thing happened. The G quadruplex disturbed, it opened up. Fluorophore cannot bind here. No fluorescence here. And then we thought like how to trap the silver. That's well known in literature. If you take a cysteine, which has sulfur has more affinity for the silver. So if you add cysteine to it, you can easily take out the silver from here because the silver has been taken out. It will again come reversely back. It can come to the same thing. It can show fluorescence. So we can make the fluorescence off, fluorescence on by adding silver and cysteine. So what basically we are trying to do is we can even detect silver and cysteine, and both things are very important. Silver is important because in the cells, many times silver is antibacterial, used as for the bacteria, right? So if it is antibacterial, many times we need to know how much is the concentration of the silver in a particular thing. So we can detect with that way. And the cysteines are also very important because our cells have biothiols, cysteine, glutathione, and the other homocysteine. These are the biothiols which play a very important role in the cell. So we can even, because this RNA has no modification, we can even express it as a gene product in the cell. We can determine presence of silver in the cell. We can determine presence of biothiols in the cell by using a very simple strategy. Very simple. If you want to do in your lab, you just need a RNA which you can order, silver you can order, cysteine you can order, all the things you can do, right? Very easy. Same with the previous project you see. If you want to detect some microRNA, you make the sequence, just change the RNA sequence as per your requirement, order the RNA, order the DFHBI, which is available at Sigma, and just detect any microRNA of your choice, right? So this is a and this is the data. This last slide. This is the data which I'm showing.
Hello, Dr.
Yeah.
Sharma. Can I just interrupt? Sorry. So since we are running very late, will you be able to finish up within 2 minutes, please?
Within 1 minute, I'll finish.
Okay.
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 Yeah.
Thank you.
So this is the we can detect the fluorescence, and then you can see this is the emission spectra and the circular. This is just showing the concept what I talked about, right? So it shows like all the data support that thing, that there's a reversible modulation happen. It can turn on, it can turn off, and then we also showed this thing in the cells also. So you can see these cells, these green things, green bacterial cells. You see, its green color is not from GFP. It's not green fluorescent protein. It's our fluorescent RNA, broccoli or spinach, right? So this green thing you see, when we add silver, it goes down. When you add cysteine, it comes up, shows the same thing, that silver and cysteine can reversibly modulate this thing. And with this, I'll end the presentation. So these are the two students who have worked. Basically, it's Rashi's work I'm presenting today, the whole thing. And Murli also helped her in her work. And I'm thankful to ISA Tirupati, to Serve India for the grant, DBT for the grant. And thank you, all of you, for patience here. Thank you so much.
Thank you for the informative speech, sir.
Okay, thank you so much.
May I now request Professor Deepthi Menon to kindly Sir, can you please stop the slide sharing?
Yeah, is that okay? Thank you.
Yeah, thank you, sir. It is understood that she is finding difficulties to join. So let us move on to the next session. So I now invite Professor Meenakshi Sundaram and Professor Upadhyay to kindly join us for the product launch of molecular biology grade reagents and chemicals. May I also invite Dr. Ramchand onto the stage, please. We also proudly announced the launch of beaded agarose and MNP agarose kit. Sir, may I please request you to launch the kit?
You might have seen all these products. Like we have three categories of products. One is all related to DNA extraction, RNA extraction, protein, size selection, like that. The other category is that nanoparticle, whether it is iron or copper or silver or gold, coated, uncoated, and stabilized. And third category is the molecular biology reagents, which probably you might be knowing why molecular biology reagents. For example, if you get sodium chloride from the shop, it will be probably 10 rupees or 5 rupees. And if you want a gram of molecular biology, sodium chloride or glucose, it should be probably 30, 40, or at least 20,000 rupees. That is the price difference because it is almost 100% pure molecular biology case. Anything what you get. The same is the case with whether it is an enzyme, polymerase, or similar things. You might have seen we have also launched agarose. There is hardly anybody is making agarose. At least 30 years back, I was involved in developing that. After that, I have no idea. Now, these are all very complex procedure. For example, many of these things are we make ourselves and all the qualities are tested. The most important thing is that the quality of the product. Now, the third category of product is like, bearded agarose. You might have heard about Cephalose. There's a series of Cephalose, Cefacryl, like that products are there. And no one is making in India Cephalose because Cephalose making, you can't call it Cephalose because it is bearded agarose. So we call it Express, Express Mag agarose beads. We will probably put a different name after some time. Now, it is basically beading agarose, and the process is not that simple at all. It took quite some time. Now, the beaded agarose, then cross-link beaded agarose. You might be knowing, those who are in molecular biology or enzyme purification will know what exactly this is used for. Mainly gel permeation and, this type of various applications are there. Now, we have also included nanoparticle embedded beaded agarose. So just I wanted to tell what are these products. Now, probably in next 1 or 2 years, we will be having about 200 to 250 molecular biology reagent. And we are not introducing all other products. For example, Sigma has got 250,000 different chemicals which is needed for research. So we are not going for that level, but about 200 to 300 different products which is used for molecular biology. So almost about 50 to 60 products are kind of ready. And next 3 to 6 months, we will be introducing the remaining products. Yeah, but then again, let me tell you, selling this is the most complex thing. is not that easy as you think. Probably it might take, even if you come to your university, probably you will think 10 times. So it's not an easy procedure, which probably some other time we can discuss. And thank you. I wanted them to talk, then they told me to talk. So
Thank you. Would you like to tell about the Peter presentation? Okay, fine.
Thank you all for joining us for this product launch. May I request Dr. Deepthi Menon to kindly continue with her presentation. Can you make it full screen, ma'am?
Yeah, it is full screen. Can you
Okay.
see it full screen now?
No, ma'am, it is still in read mode. It's still not in read mode. She has made. Can you make it again, ma'am? Can you share the screen presentation again, ma'am? Yes.
Now it's the full
You
screen.
can continue with your presentation. Yes, ma'am.
Sorry for all the trouble. Good evening. I'm Dr. Deepthi Menon from Amrita. My topic of presentation for today is fluorescent nanoparticles for biological labeling. And firstly, thanks to the organizers for considering me for this presentation. So I just want to say that I represent the Amrita School of Nanoscience and Molecular Medicine, which is a part of Amrita Vishwavidyapeetham in Kochi, and where our focus is mainly on low cost, high quality products wherein we combine basic science with translation. We also provide interdisciplinary research oriented teaching with MSc and MTech streams of different kind along with PhD. So before I begin, I thought I'll just mention to you that the size scale is what is very, very important in nanotechnology. And I'll be actually highlighting the size scale which is marked here, which is the 10 to 100 nanometer scale. And in this particular size scale, we can engineer diverse kinds of nanomaterials, which are either zero-dimensional, one-dimensional, two-dimensional, or three-dimensional. It can be based on synthetic or natural materials. And we can utilize them for diverse applications, including medicine, food and cosmetics, agriculture, and environment health, technology, and industry. Our concern for today is mainly medicine. And if you look into the global market, by 2029, it's estimated that the US global market alone will be around 60.86 billion. And now, when you consider these engineered nanomaterials, you can actually transform them for any kind of biological application by integrating different kinds of biomolecules with these nanomaterials. And you can make a hybrid and utilize them for diverse applications, ranging from therapy to diagnosis to catalysis, etc. And that brings the new terminology, which is that of nanomedicine, which introduces a new era in personalized medicine. Which
Excuse me,
actually,
Dr. Deepthi.
sorry,
Can we make sure that you are in the first line?
sorry.
Can you make sure that you are in the first line?
I didn't get you.
No, have you moved the slide or you still in the first line?
Oh, I have moved many slides.
No, but then we could see only the first line. Yes, yes. Now it is visible. Thank you.
So I was talking about nanomedicine,
One sec.
which is
Can you just
actually,
cancel your full screen and then proceed?
I'm actually in full screen. I don't understand.
No, no, you just cancel your full screen and then you proceed.
Okay. Yeah. So we are talking about nanomedicine, which actually implies the medical applications of nanotechnology, and that can actually encompass diagnostics, therapeutics, tissue engineering, and regenerative medicine. When you talk about the benefits, it's mainly the faster diagnosis, very precise diagnosis, which can target only the diseased organs and not really the healthy tissue. So, if you ask yourself why this difference with nano, I would just like to highlight that this is where the difference is. You have a cell which is micron sized, and the nano devices that we are engineering are very small, of typically sizes less than 200. So, multiple different nanoparticles can enter a particular cell, and that makes it very specific and sensitive. And as you all know, nanoparticles have diverse characteristics which are very, very important, of which the physical properties, the chemical properties, the optical properties, electrical, mechanical, magnetic, all of these are actually very, very different when the size scale changes to the nano regime. In here, I'm just going to talk about only the the optical properties, which actually encompass the light emission, and that is with quantum dots. So, what are quantum dots? They are semiconductor nanocrystalline materials of size less than typically 10 to 15 nanometers. And the optical properties of these quantum dots are actually altered by quantum confinement. What do you make these quantum dots with? With traditional semiconductor, 2,6 semiconductor materials like cadmium sulfide, cadmium selenide, cadmium telluride, zinc sulfide, zinc selenide, zinc telluride, etc., etc. So these are what you call as a conventional quantum dots. Now, the characteristic features of these quantum dots is that you can actually modulate their size and vary their emission color simply by changing the size in very, very small quantities. And it is very narrow and high intensity emission. And along the same lines, it has very high absorption. So these are characteristics of quantum dots which are well earmarked in literature. And most of the well-known materials so far in literature are all based on cadmium based materials. So cadmium selenide, sulfide, telluride, etc. And you can see the very good structure, which in the size range of 5 nanometer, and you can see the very uniform structure of each of these nanoparticles. So we can make them into very uniform size. What do you want to use it for? You can use it for bioanalytics, fixed cell imaging, for biosensors, in vivo animal targeting, ex vivo live cell imaging, etc., etc. So basically, we are trying to make use of the fluorescence from these quantum dots for diverse applications. Let us look into one of the applications, which is immunofluorescence staining. What do you do nowadays? You use fluorochrome conjugated antibody, which gives you a quantitative measurement and easy simultaneous multiplexed imaging. And most commonly used fluorescent dyes, as you all know, fluoresceins, rhodamine, cyanin dye, alexa dyes, etc. We have a big range of dyes, organic in nature. Any problem? Yes, the problem comes through its photo bleaching, which actually demands faster image acquisition, very small Stokes shift, which is basically to say that the excitation and the emission wavelengths of these fluorescent dyes overlap, which implies that you have a very high background signal which cannot be masked by the fluorescence from the organic dyes. And of course, the long-term storage is limited by the degradation of the organic fluorophores. So here is where the quantum dots have their own benefits because they have very high fluorescence intensity, broad excitation, and large Stokes shift, narrow and tunable emission, very resistance to photo bleaching, and long fluorescence lifetime. And you can see the red at the bottom panel and the green on the top panel, both are quantum dots. Whereas in the middle, you can see the green on top is actually a fluorescent dye, and the green at the bottom is a fluorescent dye. So which means even at 180 seconds, it actually fades. All these were very clearly shown way back in the 2000s. So photo bleaching of organic fluorescent dyes is a big problem. Now, additionally, concerns are also with its toxicity, because we are talking about cadmium, selenium, tellurium. So dissolution of heavy metal ions is a big concern. And very importantly, they can be developed only through stringent organometallic synthesis conditions. And there can be instability in fluorescence. So all these actually demands surface passivation of quantum dots. There are very many means of surface passivation. You can coat it with silica, you can give a polymer coating, PEC coating, and the very well studied thing is to coat it with a wide band gap material, which will actually result in a co-shell quantum dot. But again, here we are talking about cadmium based material inside and a shell which is made up of zinc sulfide. So there are materials which are already in the market, but there are some limitations. So how do we use these materials specifically for targeted applications? Simple bioconjugation. When I say simple, it sounds so simple, but it's not very simple. But you can do it very quickly using some experience with, coupling chemistries. You can actually, this interior orange shell is actually a quantum dot. You can couple it with various functional moieties on the surface. You can conjugate it through amide coupling with antibodies, etc. So bioconjugation is actually the term which is important. I just want to project this slide very importantly. This is actually the work that was done in 2005, wherein using cadmium-based quantum dots, you can see it is multiplexed imaging, simultaneous 5-color imaging in fixed human epithelial cells using quantum dot bioconjugates. You can see that the substructures in cellular proteins have been marked with different quantum dots. When I say different, it's the same material, but which emits different colors. And one very important hallmark is that all of them are excited by a single excitation wavelength. So you can stain the nucleus, the cell proliferation protein, mitochondria, microtubules, actin filaments, and whatever you want to stain. You can stain using the same material, but with different sized particles which can emit different fluorescence under a single excitation. The same material can be used in vivo as well. So all this is done. The question that we asked ourselves was, can we fabricate bio-friendly cadmium-free nanocrystals? The answer comes, yes. We can make quantum dots which are based on aqueous chemistry, zinc sulfide, zinc oxide, calcium fluoride, silicon dioxide, yttrium oxide. All of them are water soluble with a non-toxic composition, and very simple room temperature aqueous chemistry can be utilized. And the most important thing is it is size independent emission. You can use dopant ions to introduce fluorescence emission. And the same way as you did earlier, you can surface cap them with biomolecules. I'll just show you. I'm not getting into the details of that simple aqueous chemistry. We make them do it in the labs also. Zinc chloride with any dopant ion that you want and have a capping agent, react it with sodium sulfide, and you get zinc sulfide. Whatever the dopant you use here gets introduced. So I come to the result. You can see, if you have sulfur vacancy, you can get green emission. Use copper and aluminum in the zinc sulfide matrix, you can get green emission. Use manganese, you can get orange emission. So by simply changing the dopants, you can actually tune the wavelength of emission. Zinc oxide, you can use dopants like lithium, sodium, potassium. You can get very beautiful zinc oxide nanoparticles with tunable emission again. Now, you can cap any one of them with any kind of biomolecules. Call it folic acid, dextrin, BSA, streptavidin, or any antibody. You can understand whether you have done it appropriately using FTIR and other stuff. Now, here comes the point. You have the quantum dot and you have your exterior surface conjugated with whatever you have. Mind you, your particle size should be in the regime of 10 to 100 nanometer. That's the only thing which we need to target. And our aim was to target individual cancer cells using quantum dots. So this is what we expect. This is a cancer cell which has certain specific receptors that are overexpressed, and you can target them using your antibody or any other biomolecule. Here are some examples. So what we have used is KB oral cancer cell lines. You can see when you have an antibody which is conjugated anti-EGFR, which is overexpressed in KB cell lines, you can beautifully decorate these cells with these fluorescent Mn-doped zinc sulfide quantum dots. Now, if you ask me, did you check this in other cells? Yes, we did that. And did you check it without the antibody? Yes. So it is non-specific. Only with the antibody conjugated thing was it specific. So like this, multiple experiments have been done, and we have optimized the particle concentration, incubation period, all that. Let's talk one more material, yttrium. Yttrium nitrate can be reacted with sodium hydroxide, yielding you yttria, Y2O3. Use dopants like gadolinium or europium. Gadolinium can make the material magnetic. Europium can make the material fluorescent. So multifunctionality, fluorescence and magnetism together. You have particles of size less than 20 nanometer. These were works that we did quite some time back. But you can see the material, europium doped material, is highly fluorescent, and it has a fluorescence wavelength of 612 nanometer. And the material which was undoped was diamagnetic. By increasing the concentration of gadolinium that was used into the europium doped material, we introduced magnetism into the material. So we have a multifunctional material. Look at these beautiful fluorescent images of cancer cell lines which are targeted using antibody conjugated. Just wanted to show you one picture. All this would have been presented better had it been in the presentation mode. I'm sorry for that. So you can see that even with these nanoparticles decorating, this is actually 36 hours of incubation. The cells are dividing, which means that the material is non-toxic. So we are talking about a very bio-friendly material. Calcium fluoride is yet another example. Vary the fluorescence by varying the dopant. Terbium gives you green. Europium gives you red. So you can do all these. Now, antibody conjugation. I just wanted to say that you have to mind the particle size even after antibody conjugation. These are all hidden slides. I just wanted to show you that we can use it for immunofluorescence applications. This is one important thing that we did. So we took fresh frozen samples as well as formalin fixed paraffin embedded tissues from the clinics. You know that we work with Amrita Hospital, work inside the hospital. So we wanted to demonstrate the utility of our quantum dots for this specific immunofluorescence application. And we have taken fresh frozen tissue as well as FFP, acetone fixed it, deparaffinized and rehydrated, and did the antigen retrieval, blocked it using casein, and further incubated using the antibody conjugated nanoparticles and did it. Let me come to the result. Positive control was skin. So what did we use? We first did the HND, we did the immunohistochemistry to prove that there is an overexpression of EGFR. Bare zinc sulfide was used with the same sample. You can see there is no fluorescence, but EGFR conjugated zinc sulfide gave you very good fluorescence. So it's a very good fluorescence marker. Now, as negative control, we use brain samples in which EGFR was less expressed. You can see there is no fluorescence. So I just present to you the final set of data, even on the fresh frozen, as well as on the other sample. I had put it overlaid, so I'm not able to show you. So we worked with multiple patient samples, and you can see comparison was between HND, hematoxylin and eosin stained, immunohistochemistry, and immunofluorescence using EGFR conjugated zinc sulfide, which showed you the brilliant orange color, and bare zinc sulfide showed you fluorescence here and there, but not at all specific. So we have a simple technique with which we can stain immunofluorescence with on the tissue samples. Now, we wanted to prove that with multiplexing. So what did we do? We targeted the membrane receptor EGFR using one color, and we targeted the nuclear receptor Ki67 using another color. So we did sequential addition, we did cocktail addition. Finally, we understood that the cocktail addition worked. What you're seeing here is again HND staining, immunofluorescence of both together. What are you observing? EGFR, which is shown here, IHC. The orange color shows the zinc sulfide emission. Green color represents a Ki67. This is immunohistochemistry of Ki67. So we have done on one set of samples, we are showing the data which clearly tells you that we can do multiplexing. There's very simple things which you can do in the lab. Same way, we can utilize these quantum dots which emit different colors for fluorescence in situ hybridization, or FISH, direct quantum dot labeling of modified oligonucleotide nucleotide probes can be done through any receptor ligand interaction. Just giving you an example, there is a subcellular localization of the vesicular monoamine transporter mRNA. You can see that. You can see the beautiful thing. The blue color is actually your DAPI, stains the nucleus. Subcellular localization is shown in red of the mRNA. Here, you can see that multiplex labeling and multispectral FISH using quantum dots and organic fluorophore. So you can see the multiplex labeling using quantum dots, using FISH. So FISH is another technique. So like this, we can actually talk about multiple things, but what I just projected to you are two different techniques, immunofluorescence and fluorescence in situ hybridization using bioconjugated quantum dots, which can provide a unique diagnostic tool. And these nanoparticles, you needn't go with very stringent chemistry. You can make them using simple aqueous chemistry routes at room temperature. I would like to acknowledge, I know it has been a marathon talk, but acknowledgements to all my students, my collaborators, clinicians at Amrita, and all my collaborators, national and international, and even at my own center. And funding agency, not to be forgotten, for all the benevolent funding provided. Amrita for all the infrastructure. Thanks to all my gurus whose inspiration and guidance is driving me in nanobio research.
Thank you for the informative speech, ma'am. I'm delighted to introduce our next speaker, Dr. Tripti Swankar, Professor, Siksha Anusandhan University, Bhubaneswar, India, whose principal research interests are machine learning, omics data analysis, and medical image analysis. She has expertise in development of web-based tools for image analysis and deep learning of human diseases, including cancer. She'll be joining us online. May I request ma'am to address the gathering?
Yeah, very good evening to all of you.
Good evening, ma'am.
Good evening, good evening. Thanks for a nice introduction. But definitely I was hearing all the people, and maybe I'm out of box person who is from the computer science background. But definitely I love to work with the domain experts, and which I have heard over here. So let me just share my screen. Can you see my screen?
Yes, ma'am.
Yeah.
Can you make it full screen mode?
Yeah, please. Yeah, is it now visible?
Yes, ma'am, you can go.
Yeah. So very good evening to all the people, and my special thanks to Meg Genome for giving me this opportunity, and specifically to Dr. Ramchandran and Dr. Kamalika, who was coordinating all these events. And I was just hearing all the people who are really the domain expert. And as I told, I am the person not from the domain side, but from the computer science perspective. And I work on the biomedical domain. Just previous talk, I was hearing Deepti ma'am, and I was able to see in what all form the data are present. Because we as a computer science people, we play with the data. And I got knowledge in what different form the biological data are present, and how the information or knowledge can be extracted. But also understood one thing, because nowadays the data is in a huge quantity. Definitely, maybe as a computer science person using the AI techniques or machine learning, we can be a good helping hand, assisting hand to our domain experts. And thus, I love to join hand with the domain people, and really thank to Ramchandran sir to give me this opportunity to hear the domain experts. So my today talk, as I told, I'm a computer science person, so definitely, and I work in collaboration as I'm working in a university. So we have got our medical institute also. Hospital is there, most preferred hospital in the Eastern Zone. So I work in collaboration with the doctors. So my today topic is the gene selection and hub gene identification using multi-omics data and machine learning. So definitely we use data in some form, the medical data in some form, which help us to do the analysis. So no need to tell in detail about these words because almost all of you are from the life science, but just I should, for the completion of the talk. So as we all understand that genes are the segments of DNA that codes for cells to make proteins. So alteration in the gene or chromosomes alter the protein products and can hence cause some disease. So our main focus in my lab, we basically focus on the cancer related diseases. So such one of the work of my scholar today I am going to present. So as another very important thing, as a life science people, you all agree that all genes in a gene chip, because we need data to be represented in some form. So we use the NGS data or the microarray data, expression data, maybe the sequence data for our analysis purpose, or maybe the image data. So we all know that whatever the genes are present in the gene chips are not informative. So analysis of this, if it is being done by the human, it becomes very expensive. So we need some automated system which is going to make the process cost effective as well as time effective. And there comes into the picture what nowadays a lot of people are speaking about is AI, artificial intelligence or machine learning. So the limitations with these data which are present is a high dimensionality. So the number of informations, or what we call the information for us over here at genes, is very high. So we are trying to extract very informative genes among the massive data that are more predictive to class of interest. So the disease related genes we want to predict. Maybe few of the genes are well known, but still we all know that still there is a way where we can explore that what are the other related genes. It provides a vital clue for disease mechanism. It helps us to reduce the cost of clinical diagnosis, can contribute to personalized medicine. So that is the whole objective why we people as a computer science people try to design the model which can select the genes of our interest. So another thing which we all understand is disease is rarely a consequence of abnormality in a single gene. Yes, it's due to the complex interplay between the functionally related genes. And that is what is understanding the cellular interconnectedness in disease prognosis leads to identification of disease genes and disease pathways. So that is the main objective with which my lab works. So another word which we try to use, or which is being widely used when we talk about the gene selection, is nodes with relatively large number of interconnections, because we know if that node is getting disturbed, then definitely it is not a large number of other neighbor nodes also get disturbed. That is the reason it is when a person is suffering from one disease, many other problems also comes into the picture. So role of hub genes are most significant with respect to the status, survival time or age of the disease. Study analysis of hub genes are promising in understanding metastasis of disease at molecular level. So our one of the objective was to find out these hub genes. So this is one of the visualization I have given, that yellow nodes are the hub genes which are highly connected one. As my title says, multi-omics data. So you all being a life science people very well understand the omics data are present in different forms. So we targeted not to just take one type of data. Normally people work with just the transcriptomic data. So we have tried to take the data from the different labels because each one is interrelated and do our studies. So we, in the work which today I am going to present, we have considered the epigenomic as well as the transcriptomic data, and to some extent the protein-protein interaction network data also. So the limitations which we found when we are studying the single omics data is related with the data quality, because it depends on how, in what way the data has been prepared. So if it is not being prepared by a very experienced person in a very standard way, definitely the quality of data won't be good. And as the machine learning is totally dependent on data, so this may be a big challenge on the knowledge which is being discovered. Another is the dimensionality of the data, potential confounding of data, unable to discover signaling pathways when it is a single data is there. So individual analysis of single omics data lead to partial exploration of genetic etiology behind a complex state. So with that into the mind, we went into something that we not only we wanted to study the single genes which have been selected, but we wanted to bring the concept of genetic network. And that is a reason why we combined the analysis of multi-omics data, which can compensate for missing and unreliable information, which contribute towards the reduced rate of generating false positive and characterizing the genetic variation behind the complex trait. So the main objective of our work was to perform gene selection and thereafter to identify few signature hub genes, considering multiple genetic networks into account, using multiple levels of omic data, which are statistically competent and biologically enriched. So she's my one of the scholar who is already now completed her PhD, and her main focus was in this the integration of the omics data. So one of her work here I am going to share, which is related with the epigenomic and transcriptomic data analysis. So our main motivation over here was the microRNA, which is a small non-coding RNA sequencing consisting of very 20 to 30 nucleotides. And it is well known that dysregulated expression of microRNAs are associated with the induction and progression of various cancers, including oral cancer. So our main focus here was studying the oral cancer and finding the genes related with it. So through epigenetic, transcriptional, and post-transcriptional alteration, we wanted to do that thing. So that was the thing which was getting affected because of microRNA. And microRNA plays an important role in post-transcriptional regulation of mRNA expression. So that was the thing which we observed, because of which we went for this study. So exploration of microRNA, mRNA target interactions becomes significant to elucidate the mechanism by which microRNA act in carcinogenesis process. With this as an objective, we had certain study, and we came up with the thing, because nowadays in machine learning, people are talking a lot about the deep learning concept. So we wanted to include that concept and see, because our previous work we did with the traditional machine learning one, and we observed, yes, with these data also, because nowadays sufficient number of data are available, we were able to achieve that thing. So genes which are functionally related are likely to be statistically dependent and contribute to the biological outcome in synergic manner. So with this, our confined objective was identification of valuable microRNA target genes for the dysregulated microRNA that is conducive to accurately predicting the disease state. In light of this, proposed a two-step. We have proposed this graph filter based, because as I told, we are using the network based approach. So graph filter based deep learning model we designed, in relation between the genes along with the biological data at multiple omics level. So this was a small piece of work being done where this was the data set which we took from the TCGA portal. We have taken the data set, and this was the details about the data set. And these were the certain steps. So we did some type of data preparation in order to get a clean data, because we saw that it was having certain missing values. So definitely, as the model learning is totally depending on the type of the quality of the data, we went for data pre-processing. And then we did some methods were being applied to identify the significantly dysregulated microRNA, for which we use the p-value and the log fold change value. And then we, using this significant microRNAs, we tried to find out the targeted genes using the open source repository, that is the target scan database we used. And highest probability of the conserved targeting value PCT was considered for the selection of the targeted genes. In the next level, what we got was the 3149 microRNA target genes was being taken out by using the data preparation part. We pre-processed the gene expression data and expression level of those who were significant expression levels. They only were further taken for the analysis purpose, and thus we had the targeted 2,334 target genes were there. So they were being validated on the aspect of differentially expressed, disease relatedness, and predictive efficacy of the genes. This was something which we did to find out the differentially expressed genes, and the crayon colors we saw that there were the differentially expressed genes. And out of that number, that 2000 something, 331 genes we found to be which are related with either the cancer, so as well as the oral cancer and the other unknown things. So then we tried to run the predictive models on that, and we measured the different evaluation parameters. So what we observed was the integration of network information along with this transcriptomic data, because here it was just the transcriptomic data. So now we thought of integrating this transcriptomic data with that of the network information, and we tried to integrate it with the gene correlation network as well as the PPI network. And this was the thing which we did over there. So I won't go in detail of this, but definitely the same data set was being considered. Along with that, now we considered the PPI network data. So previous data, previous analysis remained with us. Along with that, we also added, in order to integrate the network information, we have taken the PPI network from this database. And these were the further steps which we added to it. So microRNA expression level of target genes was 1,348, which we, if you can see over here, we tried to generate the ensemble network of genes. So it was along with the PPI network, the gene correlation network was being somehow we integrated it, and for which we use the graph-based DNN, and we got this one. And on that part we have done, this is the detail of the model which we have used. So here the thing was, the genes connected in the resulting ensemble networks were structurally connected as well as statistically correlated. These were certain measures which we have considered while building up this network. So it puts a light on their functional association. Embedding ensemble network in the feature selector layer in graph-based DNN model establishes sparsity in the connection. So as we have embedded it using this technique, now again we got a smaller network. So it filters connections between the input layer and the first hidden layer, and a feature in the input layer can now only feed information to the immediate hidden layer neurons and correspond to the features. So by doing this process, we were further able to reduce the number of selected features. And this is something what we observed, the results of it. Modules of co-expressed genes with topological interconnectedness designed by assigning color codes. So different modules we got, that is the small networks. I should say the small connected networks, because our objective was to find the hub genes. So in order to get the hub genes, we wanted our whole gene set, whichever has been selected, to come in the form of the small networks. So that is what has been done over here, and we got certain different modules. So each color is representing the different set of genes, and the size of this band is representing the size of the graphs which we have got, or the groups which we have got. Then we did something that false positive rate and this. So we compared our model with the traditional neural network model, and we measured certain the measures like what was the precision, recall, F-score, and accuracy. So we can see that the proposed model of ours, it came up with an accuracy of...
Hi, Dr. Tripti. Sorry.
Yeah, please.
As we are running short of time, can you just complete?
Yeah, please, definitely. So these were the certain results, and I should certain genes we have named, which we got while running our model. And so the similar here, the statistical analysis we have done. I won't go in detail of this, but definitely let me conclude with these things. So the identified microRNA target genes were associated with various biological processes that lead to oncogenic transformation. This was the conclusion from the result which we got. And then we also observed that with respect to the pathways, that genes which were being selected, which was being identified using GFDNN approach, are highly associated with P13K AKT signaling, RAS signaling, which is altered in more than 30% of the oral cancer patients. So these were certain hub genes, the yellow one, which we got, and they were related with the oral cancer. All the genes were the hub genes, but they were the oral cancer related hub genes, which our model was able to find it out. So summary of my work. So proposed this approach, select the key microarray target genes with relatively high classification accuracy. So that was how we were able to extract the very informative genes, considering the two different types of omics data, that is epigenomic and transcriptomic, and embedding the networking concept. So the conclusion of this work was gene selection approaches based on integration of multi-omics data can aid the precise identification of genes specific to disease. So that was one of the thing, and this study can put a light on an alternative research direction for biological network analysis and bring to the notice of researchers for a more extensive investigation of the candidate genes, because that part we have lacked, that is the testing it in the wet lab. Certain observations which we had is biological process like apoptosis is found common in enriched list of biological works, the genes which we got, and this was the genes which we have got is more prominent. Future work, yes, clinical validation of marked genes and pathways. So definitely here we'll be needing the help of the domain experts, and we can further proceed in multiple cancers. Here we have just targeted the oral cancer. My sincere thanks to Dr. Ruchi Bhuiya, Department of Oral Pathology and Microbiology, Institute of Medical Science and Some Hospital, whose invaluable support was there in validating our findings. So this is my research group. So on the right side, they all are the doctors from our hospital. And they all, as I not only work with the omics data, I also work with the image data. So I have got the doctors from others who help me. And this is my group of the students who are working in this domain. These are certain work done. So I will just escape few of them. So this was work done in collaboration with the IIT Kharagpur, and this is the work done by my scholars. Most of them was on the image data. So it was a microscopic image data. She worked on the histopathological image data, and she worked on the mammogramic image data. Her work was on endoscopic images. And these are currently 3 of the students who are working on AMR. It's fine. So this is what is my work is. I was fortunate enough to work in collaboration with Intel. One sponsored project was there, which was on the validation of the AI model for initial screening in hospital setup that was being carried out in our hospital. And the main objective of this was to validate the AI models which are being developed in terms of quality of AI model, ease of use and productivity, and evaluation of cost factor. With this, I also invite all of you to one of the conference which is being organized by us. It is a IEEE conference on ambient intelligence in healthcare. It will be in the month of November. Thank you for your attention and giving me this opportunity to share my things with those of the domain experts. Thank you, ma'am.
Thank you very much for the informative speech, ma'am.
Yeah.
Our next speaker is Dr. Asif Khan Shanawaz, scientist, the Assistant Professor, Institute of Nanoscience and Technology, Mohali, India. He is also a visiting scientist of Australian Institute of Bioengineering and Nanotechnology, University of Queensland, Australia. He is a recipient of BRICS Young Innovator Award and DBT Indo-Australian Career Boosting Gold Fellowship. Sir is joining us online. May I now request Sir to kindly share your knowledge?
And see the presentation.
Yes, sir, we can see your presentation. You can go ahead.
So is it in full screen? You can see it's changing.
Yes, it is in full screen.
Does the slide change?
Yes, sir, it is changing.
All right, excellent. Okay, I'll just keep this away. Okay, all right. So first of all, good evening to everyone who is present in Ramad Auditorium there, and all those participants who have joined online. And first of all, I thank Dr. Priyanka and Dr. Kamalika for inviting me to give this talk, and I'm glad that they gave me this opportunity. So my topic of presentation, as you can see here, I'll be talking on plasmonic nanomaterials for photothermal therapy. And I'm a scientist at the Institute of Nanoscience and Technology, which is an autonomous institute of Department of Science and Technology. My lab at INST Mohali, as you can see here, this summarizes my lab's research interests. We work on various types of nanoparticles for biological applications. We work on drug delivery. We work on anti-cancerous, antibacterial nanomaterials. Today, we will be, discussing on these two materials. One is hydrogel, and another is a photothermal material. Both are photothermal materials, but one is a colloidal material based on iron oxide and gold, and the hydrogel is predominantly based on gold, which is encapsulated in metal organic framework based hydrogel. So prior to getting into the actual topic, we need to understand a few things about nano biointerface, which is a very critical aspect for biomedical applications with nanoparticles. And here you can see that there are, on all the four sides, you can see different properties of nanoparticles are indicated, which are very crucial for biomedical applications. Biocompatibility, of course, the material needs to be safe. That is quite quite understandable. It should not be toxic. Of course, when we come to the solubility of material, it should ideally be hydrophilic on their surface, so that something called protein corona in the once it is introduced into the biological system, the proteins that are present in the biological system doesn't absorb onto the surface. So solubility matters a lot. So for making it solubility, you need to give them proper functionalization. And the zeta potential is one of the crucial aspect of nanoparticles. If we go highly negative, because the most of the biological barriers, such as cell membrane and all the other barriers within the body, are negatively charged because of the basal lamina, which is negatively charged because of the phospholipids that are present in those sites. So what happens is there will be a repulsive force. The nanoparticles, even if it wants to interact with the biological surface, it will the negative charge on the biological surface will try to repel it. And in that case, can we have it in a positive charge on the surface? Yes, to some extent. High surface positive charge will also be toxic, because it will be too much interacting with the membrane, the basal lamina, that it will end up disrupting the cell membrane as well. So there needs to be a balance. The most ideal surface charge is no charge at all. You need to, if the material is neutral and neutral without any net charge, it is the most idealistic surface characteristic. But it is really difficult for a colloidal system to be stable without much of a charge. Yes, I mean, most of the pegylated and polymer-based materials are close to neutral. So one can, go for such kind of steric stabilization. The last one is the size. And size is very crucial, and that is where the whole talk will rely upon. Size is very crucial. Why? Because nanoparticles can be made of different shapes, and at least one of their features should be less than 100 nanometer or close to 1 to 10 nanometers, so that they can be called as nanoparticles. And however, the higher the particle size, they will get recognized by something called mononuclear phagocytic system, the MPS, which is basically macrophages which are present in various tissues. There are residential macrophages in every organ, and also there is circulating macrophages, which is the reticuloendothelial system that is present in our body. And these macrophages are going to remove any particle, any foreign material that is that comes into contact with those tissues. So anything very large in size is going to be easily recognized by the MPS system, and anything very small is going to get diffused into the body very fast. But small is quite crucial, because it is important that these materials can get cleared from the body over a period of time. When we talk about an organic material such as polymer, which can degrade to smaller pieces, which can be used up by the body, such as PLG, which can give out lactic acid and glycolic acid, which can be used by the body. But in case of inorganic materials such as gold, which is neutral and doesn't react and doesn't degrade, if we make bigger gold nanoparticles and introduce into a biological system such as our body, it's going to get retained by the body, because there is no way it can degrade and clear out of the body. So the clearance in case of renal, that is the kidney based excretion through urine, and also biliary clearance, which is the liver-based clearance through the feces, that is hepatobiliary clearance, which it decants into the stomach and it gets cleared out of the body. So specifically renal clearance plays a major role, and the recent studies are targeting towards renal clearance of various inorganic materials such as gold, where if we size them less than 6 to 8 nanometers, they can actually pass through the glomerular, capillary membrane, because the capillary has very small pores which are called fenestrae, and they have a size less than 6 to 8 nanometers. So the particles can freely move through these membrane and get into the bladder and gets cleared in the urine. However, the surface charge matters a lot as well. Neutral charged particles less than 6 to 8 nanometers are much more favorable for such clearance. But let us tackle one point at a time. Size is what is going to deal with in this presentation, not the charge. Maybe with more data in the future, we can have a much bigger data set of size, charge, and all other aspects where we can have better clearance. As I said before, gold is a material which is neutral. That's the main reason. It is made as jewel. It doesn't react with the skin like any other metal. So mostly it is neutral and doesn't cause any irritation or any kind of inflammation in the body. So gold, as I said before, can be used for making various nanoparticles of different shapes. And specifically, you can see all these shapes can be made of varying size range, as small as 10 nanometer to 200 nanometer in case of spheres, solid spheres, completely filled in solid spheres. Nano shells are hollow, shells, and nanorods, as you can see, they are, elongated structures. Nanoclusters are not exactly bigger particles; they are atomic clusters, and you can count the number of atoms that are made that make these nanoclusters. Basically, they are 1 to 3 nanometers in size, and these are much favorable materials for for in vivo application, because they are less than 6 to 8 nanometer, which I indicated in the last slide. So they get cleared, and there are so much of work that is done in the literature, and we are also working on it. And nano cages and nano stars are also, up to 200 nanometer they can be made, but they are all more than 8 nanometer except for nanoclusters, and they will be really difficult to get cleared out of the body. So we wondered if we can make materials which are 6 to 8 nanometers or less in size. They are assembled in a particular way that they can have bigger functionality and they can get cleared out of the body over a period of time. So the first material that we will look is iron oxide nano assembly. These are very small particles of iron oxide, less than 6 nanometers. And on the surface of this assembly of iron oxide nanoparticles, on the surface you see we have gold nanoparticles. Again, they are also close to 3 to 5 nanometers in size, and they are individually, each particles are ideal for the clearance. And as an assembly, they have much better functionality, and we will come to that. And we use these materials for radio sensitization of cancer cells, and also for imaging with iron oxide, be used for magnetic resonance imaging. One of the talk covered how exactly it is done. And another type of imaging, optical imaging, is photoacoustic imaging. And we did make these nano assemblies. As you can see, these are iron oxide nano assembly, which are functionalized with polyethylene imine, which is a strongly positively charged polymer. And we functionalize the surface of these iron oxide assemblies with negatively charged gold nanoparticles, which are close to 3.5 nanometer on an average. And the individual crystallites that are making these iron oxide nano assemblies are about 6 nanometers. And overall, this is the transmission electron microscope image of these nano assemblies of iron oxide and the gold. Now, you may wonder how these materials are functional, and especially gold nanoparticles. They have something called surface plasmon resonance when they are irradiated with photons. Okay, and photons or light, which photons that is making the light, is, as you all know, is an electromagnetic radiation. and it has a magnetic component in it. So when gold nanoparticles are irradiated with this electromagnetic light, the the electrical component of these of this light is going to polarize the electrons that are present within the metal. Metal is conductive because of the free electrons. So gold is also conductive, and it is going to polarize or bring all the electrons to its surface. And as you can see here, the electric field being a sinusoidal field, it as soon as when the field flips 180 degrees, you can see the pole polarization also flips 180 degree. And because this polarization flip in the dipole, the frequency of the flip, if it matches with the incoming frequency, it absorbs that particular frequency at a larger amount. The energy is absorbed, and that energy, it is actually dissipated as a thermal energy. We will come to that. That is called photothermal energy, and we will use that for therapy. But one very crucial thing you need to understand is, in a sphere, as you can see here, in a sphere, isolated sphere, just take a nanoparticle which is spherical in shape, you can see that it is a symmetrical structure. You cut sphere into two hemisphere, you cut it on either any of the direction, it is going to give you a two equally halves hemisphere. And that means that it doesn't, such a symmetric sphere is going to give a surface plasmon resonance in the visible region. I have indicated it in green color, visible region. But when we group these small spheres into elongated thread, okay, uh, so the electron is going to, it is no more symmetrical. If you cut in this axis, it is going to give, two rods, top and bottom. And if you cut at the center like this, it is going to give 1.5 spheres this side and 1.5 spheres. So this is not symmetrical, splitting that you would experience. So that way, what happens is the surface plasmon resonance also is non-symmetrical. It has two types. One is transverse, resonance, and longitudinal reference. And transverse is the usual green band. You will still see it, but the longitudinal reference will be a new band that you will see in the near infrared region of.
Excuse me, sir. Sorry to interrupt. Can you please wrap it up because we are running short of time.
Yeah, how much time do you have?
In couple of minutes, 2 to 3 minutes, probably.
Very short. I'll try to wrap it up. So the near infrared absorption is what we are looking at for photothermal therapy. And here, as you can see, these circles show these strings of nanoparticles which provide something called which provide a broad absorbance in this region, where you see 700 to 800, which is the near infrared region, and which is absent in individual isolated gold nanoparticles. And this way we can actually assemble these materials to give very strong near infrared absorbance, which will allow in the presence of light, which will allow them to give this temperature rise. And the iron oxide nano assemblies also have very strong magnetic moment for magnetic resonance imaging. And these materials can over the period degrade in acidic condition, and you can see up to 21 days they degrade in the acidic buffer, and from as big as 200 nanometers to close to 110 nanometer, which is a hydrodynamic size, but actual size is about 6 nanometers. And you can see here, these are gold being a high atomic number metal. It can actually be used for radiosensitization. And here, these data basically indicates that in the presence of gamma radiation, these materials can sensitize the cancer cells and bring down its viability. And there are so many controls. You just look at these panels where the nuclei is quite heavily damaged because of the nanoparticle mediated radiosensitization. And these nano assemblies, as I said before, can be used for magnetic resonance imaging with a very good relaxivity of over 200 per centimeter per per millimolar value, which is quite close to the standard state of the art MRI contrast agent, and also it can be used for photoacoustic imaging, which basically is the photon when where the thermal energy which is dissipated out, which will also give out an acoustic energy which can be captured by specialized detector, and we can pick up signal from deep tissues. And because these materials can degrade over a period of time, you can see let us look only at the gold here. We can see that the gold, it gets it was it was sequestered by spleen and liver, but also it eventually decreases over a period of time, and also in the kidney it decreases. That means over a period of 1 to 28 days it gets cleared out through the kidney and the liver. Now, the second part is, within a few, minutes, I will just finish this. It is also a degradable material where it is made up of metal organic framework, and metal organic framework and polymer, they form a supramolecular hydrogel. And these metal organic framework particles are highly porous materials. They are used for gas absorption, primarily, but now it is coming for drug delivery applications. And in this work, we try to load these metal organic frameworks with very small 5 nanometer or less, gold nanoparticles. And the principle remains the same. Very closely, placed gold nanoparticles will absorb in near infrared region, which will make them, active in near infrared region. And some of these MOFs are also loaded with curcumin, and you can see how the curcumin is loaded. This suspension is made up of just metal organic framework loaded with curcumin. Now, these materials, you can see the pore size of these morph structures decreases as we load these gold nanoparticles inside them. Their surface area also decreases because of the gold nanoparticle. That means it is now occupied with these gold nanoparticles. And when these plasmonic morph is mixed with polymer, they supramolecularly assemble to form these hydrogels. And these hydrogels are self-healing. That means they can inject, they are injectable. They can be injected as and when required to whatever the site that we want it to be injected into. And very little bit in vitro, where we can show, we have shown that these materials in combination with curcumin and the gold based photothermal therapy was synergistic in action, and only in this combination we could kill as many cancer cells as possible. And in vivo studies, we directly injected these hydrogel into the tumor, and we radiated with the laser, wherein it release the
Sorry,
core.
sir. Could you please conclude, sir?
I'm finishing. So it increases the temperature, and also it releases the drug locally to form to give better therapeutic action. And the gold could also be seen in the urine over a period of time, allowing it to degrade over a period of time. And in summary, we could actually engineer degradable material which disintegrate into smaller particles and clear over a period of time. And such kind of degradable inorganic assemblies and other hydrogels can be a very good tool for in vivo application. And I acknowledge my students here and all my collaborators, the funding agencies, and I thank all for listening to this talk patient. Thank you.
Thank you for the informative speech, sir. I'm elated to invite our next speaker, Dr. Manoj N. Krishnan, Head Pharma Therapeutics, Manufacturing Platforms, Science and Business Development, Manus Bio, US. He has more than 10 years of experience in the field of drug discovery and target discovery of immune mediated diseases, and more than 20 years of experience in immunology, host pathogen interaction, and biophysician studies of potential drug targets. Sir is joining us online. May I now request Sir to please address the gathering? Sir, can you please unmute your audio?
Can you hear me now?
Yes, we can hear you, sir.
Okay, I'm trying to figure out how to share my slide. How do I share the screen on this? I can't see an option.
Sir, can you see a slightly tilted arrow in the bottom of your screen?
Yes. Okay, I see. Yeah.
Yeah.
Okay, got it now. Thank you. Let me see.
Click the screen share and go for launch.
Can you see it now?
Yes, we can see your slides, sir. Can you please put in full screen mode?
Okay, 1 second.
Yes, the slide show otherwise. From current slide. Can you put it from beginning? Can you click on the first option? Cancel it, sir. Yes.
Can you? Is it changing the slide?
No.
The slide. Okay.
No, sir. Can you do it again?
Okay. So it didn't change, right?
No, it didn't change.
Okay, I've seen that happening many times with this tool. Maybe I'll just go like this.
Yeah, you can.
Is that okay? Okay. Well, first of all, thank you, Dr. Ramchand and the larger Maggenum team for inviting me to share your joy of launching new products. And also congratulations to the Maggenum team for taking the bold step of supporting an opportunity where your expertise and drive could be commercialized and launch a venture. I've seen Maggenum over the last, I would say, 7 years, establishing, growing nationally, and I know I am witnessing that it's also growing internationally. And I'm pretty sure that in a certain number of years, Maggenum will have a significant presence on the projected bioeconomy vision of the Department of Biotechnology of the Government of India. And I wish you all the best in that. I'm also hoping that in your version 2, version 3 phase of the growth of Maggenum, you would think about tangentially innovating and finding or exploring the application of magnetic nanoparticles or even non-magnetic for additional portfolio of biomedical applications to enable larger biomedical communities needs so that the discoveries can happen faster, bench to bedside translation can be achieved faster. In that line, in my today's little talk, I don't intend to share any kind of experimental data because I don't work on synthetic nanomaterials. I work in the precision medicine space, and I wanted to bring to your attention some of the challenges at a conceptual level, which I hope that you would think in your version 2 growth to create a new class of products to help people working in the precision medicine space. So I want to mainly highlight 1 or 2 examples on the gaps where you could fill in. So what do I mean by precision medicine? So this slide shows how is the pharma going to look in 20 plus years. On the left side, it lists 5 disruptive forces that are going to define the biomedical or the pharma space in 2040 onwards, or around that period. Of that, number 2, 3, and 5 includes customized therapeutic treatments, delivering in a precise interventional manner, as well as having the ability to deliver curative effects, right? So when we have the ability to intervene with precision in a customized, which means patient-wise manner, with the curative effect, what's going to happen to the diseases? As you see on the right-hand side, the distribution of the diseases will have a very different portfolio compared to today. In 20 to 40 years, we might not have this large prevalent disease groups, but rather we will have small groups of patients in different segments. One of the effects of this precision curative therapies is that a lot of the diseases will become curable. Whether we cure or not is a different issue, but we will have the ability to cure. As shown in this yellowish circle on the left side of the right side graph, are the diseases that we will have the ability to cure in next 20 or so years. As you can see, if you look carefully, nearly all of them are caused by either a monogenic variation, or they all have a readily identified or a strongly identifiable genetic association. Some kind of a variant has been identified to correlate with the disease onset. So we could target that gene or the adjacent nodes as a therapeutic drug. And seen on the right side are the other diseases, which, with the ability to understand more and more about the genetics and the biology behind those diseases, will shift more towards the left with the ability to intervene with precision medicine. Now, if we are going to have the ability to deliver customized precision curative therapeutics, what are the tools or medicines we have at our hand right now? In this slide, I have attempted to classify all the available human drugs into 6 larger buckets, ranging from small molecules, nucleic acids, biologics, gene therapy, cell therapy, and living therapeutics, right? So all the drugs that we have today would fall into one of these groups, or some are even falling into more than one groups. So if we take a more detailed, granular look at these drug classes, we can see that some of these drug classes actually have same underlying biological concept. For example, if you look at gene therapy, what is gene therapy? We want to change something in the DNA or RNA, depending on what level we apply. And then we need some kind of a tool, which we call medicine. That can be a drug that can be used to bring the change in the genome or gene expression. Oftentimes, we achieve those changes in the genome through gene therapy by using some kind of a nucleic acid-based tool. Same thing for cell therapy. Most of the newly emerging cell therapy modalities include cells that are modified and then put back in the host. When we say we modify a cell, oftentimes we're talking about modifying the genome of the cell to increase or decrease expression of something or have a different ability. Now, if you look at the nucleic acid-based therapeutics, what is it? Again, we are delivering a set of nucleic acid-based tools that'll go and modulate the expression or functioning of a gene. So, in essence, nucleic acid therapeutics, gene therapy, cell therapy are same thing in one way or other way, doing the same thing, right? So with that in mind, it looks like nucleic acid-based therapy or tools will become the thing of tomorrow. So let's take a more detailed look at how does that field look like. Shown in this graph on the x-axis is the increasing innovation plotted against the market size of each nucleic acid modality on the y-axis. As you can see that historically, ASO, antisense oligo, and siRNA are the only 2 approved drugs up until 2 or 3 years ago from this segment. Now, we know that mRNA over the last years become the most heard household word.
Sir, excuse me. Sir, you're muted. Can you unmute yourself? Excuse me, sir. Sir, can you hear us? You're not audible, sir. Can you please check that? Can you unmute yourself? Host can also do that. You can mute.
Your audio device has some problem. Reconnect audio. If you reconnect the audio device. Otherwise, call from your logging in. Take in the presenter again. Can you speak? Can you hear us? Unmute. Unmute. Can you speak?
Hear me now?
Yeah. Please go ahead.
I think, yeah, I have. Anyway, sorry for the disruption. I think I have, what, 6 minutes? Anyway, so I want to highlight what are the challenges in this field where Maggenome or similar companies can help us. Just list some of the grand challenges in the nucleic acid therapeutics. It includes achieving highly efficient genetic change. If you want to edit something, we want to get a high editing efficiency. There's another issue, right? Oftentimes when we deliver a drug using a vehicle, there will be a immune response against the vehicle. We want to escape that. And then we should be able to do a targeted delivery with increased prolonged bioavailability. We should have a tunability, and we should also have an upstream ability to purify the delivery vehicles at scale the way we want. So I'm going to highlight the first two where nanotechnology could potentially help us. So historically, nanoparticles have been used as delivery vehicles or as some kind of a display platform for a high density accumulation of a molecule of interest for delivering or displaying whatever it is, or as a tool for imaging or sporting some kind of a cellular or tissue level information. But what I want while going towards the future is, can we take nanoparticles or nanotechnology to a subcellular level or at a molecular level where we can create intracellular platforms where precision medicine can be assembled and do its job, like it's putting a table inside the nucleus or cytoplasm, where we can get the nanoparticle to bring precision medicine tools as well as the cellular targets, and then let the medicine do the job at a platform. And also, can we use it to trick the immune system so that redosing of the nanotechnology or the gene therapy, cell therapy delivery vehicles is much more feasible. To zoom more into more details, I'm calling out one example. Those of you who work in the field of CRISPR genome editing knows that one of the ways the genome editing happens is HDR-based editing, which is homology directed repair, where we will have the ability to precisely introduce or take out a segment of the genome. The problem now is that, as shown on the left side, the efficiency of this approach is much weaker, like 1 to 5%. Could we have some kind of a nano sponge or a platform, especially using magnetic nanoparticles, where we could deliver that, loaded with a couple of the tools we need? Could be a template, some of the enzymes that mediate the process. Then once it goes into the cell, it could get all the required components for this process into one place. That may be one place where innovations can help people in the precision medicine. And the second example, I'll take 2 more minutes. The second example is the challenges in the targeted delivery of the precision medicine therapeutics. Oftentimes, we deliver nucleic acid-based therapeutics, be it gene therapy or not, through one of the other means, right? A lipid nanoparticle, which is more an artificial one, or using viruses or virus-like particles, or the upcoming modalities like exosomes, viral capsids, or bacterial needles, right? You can see on the right side, they all have the ability to wrap around nucleic acids. The problem is that you can only inject this into our body once. Once we do that, there will be a profound immune response created. So if you try to redose the same patient with the exact same vehicle, there will be a neutralizing antibody response almost certainly. And also there's another context. Many of these tools that we use for gene therapy, gene delivery, like viral particles, there may be pre-existing immunity in our body against these viruses. For example, adeno-associated virus, which naturally infect us, right? There may be an immunity in me preventing me from getting certain type of AAV gene therapy. So with that information in our mind, the major challenge here is that there could be an antibody response against these vehicles. We should have the ability to escape or neutralize or deflect those antibody response. That is one way where nanotechnology could help, bring some kind of a shield by which we could protect the vehicle from being exposed to neutralizing antibody. The other issue is that, particularly in the case of AAV, when we inject AAV into our body, 70 to 80% goes non-specifically to liver as waste, incurring a lot of cost. Only a small amount of injected AAV actually reaches our intended target. So maybe there could be a nanotechnology-based shield that could prevent liver from removing all the injected viral vectors. So that's another area where nanotechnology could help us innovate precision medicine much faster. So with that, I conclude my little talk, and I'm hoping that Mark Genome will be able to create a version to thinking beyond the type of portfolio which you are doing while innovating what you're doing too. Thank you.
Thank you, sir, for joining us. Like to invite our next speaker, Dr. Amitabha Chowdhury, Scientific Advisor, Cygenome Labs, San Francisco, who has over 20 years of experience in the area of drug discovery and technology development. As the Vice President of R&D at MedGenome, he leveraged NGS technologies to investigate diseases at a molecular level to develop insights that can lead to better therapies. I request Sir to kindly share his talk. Sir, can you unmute yourself? No, we can't hear. Sir, we can't hear you. Mm.
So we can't hear you, and we can see you. Mute and unmute and mute. Mute and then unmute. 1 minute. We'll mute and unmute again from our side one more time. Still, we can't hear you. One. No, still, sir, we can't hear you. 1 minute. We'll just check. We're checking from our end. Yes. If you do talk with audio, then make it. If it is not happening, make it. Can you speak? Oh, we still make him that other option you try. No, sir, not still. Yeah, there is some error at your end. From here, also it is showing in your audio. You already did, no? You already did. Okay.
But we just can't hear you. Okay. I am putting it on a speaker.
Shall I start?
You understand?
Yes.
Yeah. Sir, can we just start with your presentation?
Hello everyone. Good evening in Chennai. In the morning here. Sorry for all the difficulties in connecting me, but I really thank the Maximum team, Dr. Ramchan, and the greater part of the team for giving this opportunity. And I work in the US. I'm trying to the exciting products that Maximum has created. We're trying to introduce them to the US market. It's familiar with the products and the team. So I just wanted to spend a little time just introduce some of the new areas where this magnetic network is going to be used. So just as an outline, give you some introduction of NPs which you have heard throughout the day, and then discuss a little bit about Maginome's technology. And then go into the area where Maginome is trying to build the second generation of product in the area of proteomics. So you can all hear me, right?
Yes, sir. We can hear you.
Okay. So as you know, the nanoparticles, they come in a variety of different forms, like the inorganic form, which is the magnetic nanoparticle of Maxgenome, which is iron oxide nanoparticle. They have a lot of positive aspects, but they're insoluble and they can create toxicity. Then comes the polymeric form of nanoparticles. You can precisely control the particle size, but they also have aggregation and toxicity issues. And finally, there are lipid-based particles with a range of different types of properties, but it's very difficult to encapsulate payloads into them effectively. And on the right, you can see that the nanoparticles are being used in a variety of different diagnostics and therapeutic use, as Manoj mentioned, from genome editing to generating CAR T cells to delivering payloads to different regions of our body. These nanoparticles are being used extensively. Now, if you look at the Maxgenome technology, the exciting part of Maxgenome technology, and again, you have heard of this throughout the day, I'm sure. But first, besides being patented, the bare magnetic nanoparticles, which is the core technology of Maxgenome, actually results in a much better process, which means that the strong magnetic property of the beads are fully utilized, not only in binding to the target molecules, but also in binding to the magnet, making the process much more easier and much more efficient. And then, of course, there is cost effectiveness, there is automation, and we are working with a few clients in the US who are actually testing the Maxgenome beads in their automation machines. And of course, it's fast, and most important is that the technology is green, like because it is not coated. you're not using chemicals and some of those chemicals that are used for coating, and therefore the particles are biocompatible and non-hazardous. Now, this is the basic nanoparticle that MagGenome makes. Now it's available for commercialization. Now, with this magnetic nanoparticles, MagGenome has created products. So I'm talking about the products that are currently created by MagGenome, and they encompass something new, like a word that has come into the realm of health and biological sciences. It's called One Health, which is a health that comprises of human, environmental, animal. And I think MagGenome product has impacted all these areas significantly. And I think the most laudable impact of MagGenome product has been in delivering more than 25 million COVID tests, throughout India and also outside India during the COVID times. And this is all based on the bare magnetic nanoparticles that I'm showing you on the right. The second important product that is getting a lot of traction in the US is the size selection beads that are used for next generation sequencing on the Illumina platform, just shown here. And then also the size selection beads for the nanopore technology, which is to purify high molecular weight DNA, then that can go into this long read sequencing technology. Now, besides this, MagGenome products are used for a variety of different areas, from extracting DNA from plants, from soil, from human fecal and stool materials, from animals, just to create the metagenomic landscape that everyone is very interested in. So these products are being currently used, and they work very well. And these are the products that we are introducing in the US. Now the question is, where should we look for new products using magnetic nanoparticles? Manoj mentioned areas that are maybe much futuristic, but this is much more something that we can achieve very quickly. So one of the areas where magnetic nanoparticles have become of value is to isolate proteins from biological systems. Now, these magnetic nanoparticles, they can absorb proteins on their surface, and that is called a protein corona, which was coined in 2007. And over the years, with the development of mass spec technology, magnetic nanoparticle-based protein isolation and characterization has become, very not only popular, but is also, reaching a phase where it can have a tremendous clinical utility. And these, and coming to on the bottom, you can see that these magnetic nanoparticle-based proteomic approaches can be applied not only for new therapy design, but also for proteomics, like blood proteomics, identifying biomarkers in the blood, and also for ecological and environmental, studies. So here, the important, area of growth would be to create magnetic nanoparticles and modify them in a way that they can actually bind, differentially different types of proteins. And MagGenome has already initiated, some R&D work in that direction. But this is an area that I think is going to grow in the near future. Now, this is also an area where I think, MNP developers will find a lot of utility, is basically to use AI on some of the, or thousands and hundreds of thousands of conditions that people have actually used in their R&D work to generate very specific type of MNPs. And I think this AI can also be, brought in order to develop new processes to make magnetic nanoparticles of desired properties. Now, coming to one of the areas or one of the companies in the US, the Seer, which has developed this proteograph technology. So they have actually created nanoparticles which have different surface properties. And using the different properties of the nanoparticles, they can bind different types of proteins from blood plasma. And this is taken from that paper published in PNAS. And you can see on the far left, the red one is from a Seer proteograph. And you can see that by using magnetic nanoparticles, they use 5 nanoparticles. You can actually, identify almost 1,700 different protein groups compared to half or less than half by conventional methods. So this is a very interesting area that will have a tremendous impact in health, human health, and in animal health. Is to deep capture proteins and look at proteins which are much rarer and not much more prevalent. So this is the MagGenome technology of immobilizing proteins. I'm sure someone must have discussed this in the presentation. But the very interesting aspect of this technology is that the protein secondary and tertiary structure is preserved when they are entrapped within the MNP molecules, as shown here. And this is a patented technology. There is a paper that anyone can read to get more information about the technology. So the first thing I wanted to just show you two data that the R&D team has generated. One is showing that the proteins that are entrapped in magnetic nanoparticles, the HRP enzyme, it's fully functional compared to HRP enzyme that is so unstable if you just store them over a period of time. The second, or the figure at the bottom, basically shows that if you make protein A beads and immobilize them on MNPs, these beads can be reused at least 20 times. And that is, the graph shows the binding of immunoglobulin from human plasma. And you can see the heavy and the light chain. So this also, brings this very important point that by reusing beads, you are actually reducing the cost of your process, and that's so important. This is just my last slide showing that the MNPs in biomedicine is being used much more profoundly in different areas of human health, animal health, and this will continue to grow. And this is an area I think MagGenome technology leverage to bring new products in the future. And so, just as a conclusion, this technology that is being created using magnetic nanoparticles is impacting One Health products, not only in India, but also globally. And then the applications of MNPs in proteomics is an area that will evolve rapidly. And finally, AI will be increasingly used in generating, NPs that can then be used for a variety of different, diagnostic and therapeutic purposes. Thank you, everyone, and congratulations to MagGenome for launching the new products. And and thanks for listening.
Thank you for the enlightening speech, sir. Our next speaker is Dr. Aniruddha Bhatti, Senior Scientist and Head of Technical Operations, Nano Technologies Private Limited, Chennai. He is an experienced researcher with a demonstrated history of working in the biotechnology industry with skills in nanotechnology, nucleic acid extraction, and protein purification. I now request you to address the audience, sir.
Good evening, one and all. People present here and joining from all across the world over by online mode. So I am going to present, you have witnessed several talks since morning, and then most of them covered how various products, technologies which are developed in academia, technologies which are developed in industries, how translational research can be done in nanotechnology is very important. And we through MacGenome have done a lot of academic research initially, and then transformed those research into successful commercial products. And some of this, so I have divided the talk quickly, 5 minutes initially, the products which we have launched based on magnetic nanoparticles, which is our core. And the next part, one example, how in real world nanotechnology made an impact. So this is the hero of our story, magnetic nanoparticles. We use iron oxide magnetic nanoparticles, bare magnetic nanoparticles. The size is about 10 to 12 nanometer. And there are various surface properties of this magnetic particle which make them unique. And then this gives a lot of advantages in biological processes and other industrial processes. So what are the advantages? Magnetic nanoparticles offers unique surface property throughout. Today we have seen different particles having different properties. Iron oxide also give a lot of good properties which can be harnessed for industrial application. Number one, for example, in a lot of biological laboratories, you use centrifuge to spin down. When a magnetic nanoparticle is used, there is no need to spin down a sample. So it saves time, it saves cost. Then magnetic nanoparticles can be used for, it is cost effective. One very important feature, if you see the title of the talk, was automation. Automation in a lot of biological processes is only possible by introduction of magnetic nanoparticles, and then that has transformed diagnostics, protein purification, and several fields. During the course of this presentation, I'll be able to show you some examples. It is green technology because a lot of plastic waste which is generated otherwise in biological processes can be avoided here. So what we did in MacGenome, the genesis you have already listened to, we initially, today, the products that we are presenting, 2 patents make all these technologies possible. One is a reversible binding of nucleic acid, that is DNA or RNA, on the surface of magnetic particles. So this is the patent and the publication for it, granted patent. And the next one is our granted patent application for immobilizing functional protein on the surface of bare magnetic nanoparticle. So these were the two technologies which has gave way to several commercially successful products. These are the product lines. So Express DNA, Express RNA, COVID kits, Express Affinity, MagStable, and Express Pure. All these products have come because of the research work which was done and patented by MacGenome. In the Express DNA and RNA, several biological samples are used. Genomics has been the mainstay of biological science research in the last 25 years. So we have products very efficient because they are all based on bare magnetic nanoparticles from various biological samples like blood, tissue, bacteria, saliva, cell line. Same way for RNA. Another very interesting product is biological sample collection is also very important. So we have a saliva self-collection device where you can just spit and then the saliva is stable at room temperature for up to 2 years. And the samples can be transported anywhere. So this also gives a lot of impetus to direct to consumer genetic testing. How is this technology different? So there are a lot of technologies available with column kit. Some examples are given. There are some other kits available in the market which are coated beads. The differentiation that we get, why our product is better, cost effective, time effective, is because we have developed a strong IP and the particle gives the advantages where we have a distinction in IP, we have a distinction in performance, and therefore, and the cost and time effectiveness is there. So this is the unique proposition. COVID came. I'm going to give some examples for COVID later as well. But in the COVID times, within 3 months, our team was able to develop all the products which were related to COVID. So there was some saliva, DNA, RNA extraction work that we were doing. Then the pandemic came and we thought, let's channelize this. So this is also very important. Whatever research you are doing, when the opportunity arises in the market, you have to change your strategy and bring about products that can impact when the need of the hour is there. So we developed a molecular transport medium, manual viral RNA extraction kit, and automated viral RNA extraction kit. Some other products like VTM and RT-PCR kit, because that was a need of the hour, we developed. Dr. Amit just mentioned about the express affinity. So the functional proteins like affinity ligands, protein A, protein G, glutathione, can be immobilized on the surface of magnetic nanoparticles, and some protein purification based application, antibody purification, immunoprecipitation, chromatin IP, these all can be done very efficiently. Right now, how they are done? Through a column. With the magnetic particles, how they can be done? Even in a small vial, or you can scale it up to a higher volume as well. No need to set up the column. So this is where a distinction comes. How the biological process was already done can be completely transformed in an automated way. So this is how the technological advancement comes into picture, and therefore nanotechnology comes into picture. How the processes were done earlier, and they can be improved by efficiency, time effectiveness, cost effectiveness. The product which definitely we believe will be very important in the times to come. Next generation sequencing, most of the genomics big companies in India are genomics companies. In genomic workflow, for long read and short read sequencing, you need to clean up during library preparation, you need to clean up the library. You have to let go of linkers, select a specific size. So with that, nanoparticle is very important, and they can be used very efficiently. So we have a very good product which is already in the market, which is based on this. We have introduced both in India and US and working very well. Again, based on nanotechnology, a technological advancement. And finally, we realized in the COVID times, I'll come back to that in the next part of the presentation. Now we have automated kits which are automation ready on our own automated instruments, as well as on other open magnetic particle based systems which are already available in the market. So this is was the short introduction of some of the products that we have. In addition to that, for the diagnostic markets, we have the cfDNA kit, we have the FFP DNA isolation kit, we have several other products coming on. Even for non-genomics based application, we have wastewater purification coming up. So a lot of different applications are there: environmental, industrial, biological. There is a full spectrum is there. In the morning, in the inaugural session, there was some discussion about how nanotechnology or the technology coming up can impact. I just want to give you a short example with our existence, our small beginning, how we were able to help in a cause. Now, there has been a talk of biotechnology coming as a boom in the last 50 years. Definitely it came. But last 20 years, nanotechnology was also quipped as the next, blue-eyed boy, that this will change the world. It has happened, and we probably have realized or not realized. Let us see. So automation, why automation is important? The topic was automation. Why automation is important? Initially, when the COVID testing started in India, now in India, we have done close to 800 or 850 million tests, COVID tests. When COVID started, now most of the people know there was a shortage of iron extraction kit that impacted initially, imports were not possible. Make in India was just happening, and we were not able to produce enough that we can use. There was another problem, technological problem, which as an industry, as nanotechnology industry, a lot of us and some of our other companies were able to solve. How? COVID-19 extraction, now everybody knows how it is done. So nasopharyngeal swab collection, then RNA extraction, and finally RT-PCR. Think about this. The sample collection is done the same way today how it was done 3 years back. RT-PCR was a well-known technology. Maybe it was not, it has not become a household name as it is now, but people who were doing RT-PCR 10 years back, it is the same way. The master makes the plates, it was done. What happened initially? You can do through a column-based method, this COVID extraction. How many? About 45 to 50 minutes, 8 samples. RT-PCR, you need 96 samples if you have to, be cost effective. Now, imagine this. If we were doing COVID extraction only by using a column technology, how many deaths would have happened? India's strategy was divided into 2 parts. We were able to diagnose well, and later we were able to, the vaccination program. Vaccination program gets a lot of mention. But this automation that we were able to do with RNA extraction has actually changed, because now, towards the middle of the pandemic, when we became self-sufficient in diagnostics, we were able to do 96 samples in about 12 to 15 to 18 minutes. From 8 samples in an hour to 96 samples in 15 minutes. And that enabled the labs, use of magnetic nanoparticles, enabled the lab, that the way companies actually came out with a lot of automation in this time. And why automation was possible? Because of the magnetic nanoparticles, which was able to change a column technology within 3 months or 4 months to automation. So now this is a change. Includes two things: make in India and advancement in nanotechnology. So basically, we didn't realize, or if we did, it was not said enough. One technological advancement was able to save a lot of lives because we were able to identify, diagnose, quarantine. And that led to control in infection. And India did very well as compared to the other Western countries. It was speculated we might not do well because of several models and publications, but we actually did well. Kudos to all the scientists who have worked tirelessly in the COVID times. So with this, with the help of our expertise as well, we were able to do about 25 million tests, about 4 to 6% of the total market, because we were able to do make in India. All the different instruments which were pumped in India, we were able to automate our kits, and we also had our equipments. Now, if you imagine, we are installing equipments in remotest part of India. So this technological advancement just took 2 years. Nanotechnology based technological advancement just took 2 years to come and change the way how testing is done. This is one example where transformation, an idea just into the product, and how it was able to change lives and impact life of all of us. Now, so these are all the different automated equipments where we did automate our kits. What is the current initiative? So I showed you two publications. One was for the DNA, RNA reversible binding. One was for the affinity ligand. So our next initiative, in addition to these molecular diagnostic products, is making automation with the help of these magnetic particles for research domain, DNA, RNA extraction, rapid for our own equipment and other automated equipments, and also therapeutic protein purification. The protein product that we have with protein A and protein G, eventually making it therapeutic grade with regulatory approvals, of course, and then eventually having an equipment like ACTA, where protein purification can be done at a rapid rate. Right. Last but not the least, about 10 to 12 years back, I was introduced to the world of magnetic nanoparticles, and the two people who introduced me to nanotechnology, or nanotechnology became something I pursued in the last 10, 12 years of my life. Fortunately, both of them are here. Don't get confused, they all look the same, but this picture is from a few years ago. Why? Because when I started this paper, which we already in the morning it was discussed, the direct binding of protein to magnetic nanoparticles actually paved a way to lot of researchers, my predecessors, us, our team, and we have read these publications like Bible, and then eventually this paved the way. So again, simple research, applicable research, translatable research, how it led the way, and how the whole field was changed was so I just wanted to acknowledge Dr. C.N. Ramchan and Dr. R.V. Upadhyay for introducing me to this team. I found, I am missing a lot of current, but I found one picture. Our team, MagGenome team, our R&D team, our current colleagues, some people who are not with us, they are all needs a lot of credit and applause. Several hours, several years of hard work has actually made this possible. And one picture I think he talked about in the morning, Vibrant Gujarat. Somehow, we were able to find it and put it here. So thank you, Dr. Ramchan. Thank you, Dr. Upadhyay, and thanks everybody. Thank you.
Thank you for your illustrative words, sir. I would like to call Dr. Shejal Shah onto the stage to receive the memento. I also request Dr. C.M. Ramchan to present the memento. Can we have you on the stage, ma'am? I also request Professor Upadhyay sir to present the memento. May I now request Dr. C. N. Ramchan, co-founder, CEO and CSO, Magino and Terragen Group of Companies, to deliver the concluding remarks.
Thank you very much. And I have a lot of things to talk, but I am not going to do that. So let me finish in 2-3 minutes. Um, the moment if I start talking, because in concluding remarks, what you have to do is that we'll have to just brief all these 20 or 25 speakers, what they have talked here, and we don't have the time. And I'm extremely thankful to the last 30 people who are sitting here. I just counted, and about 70-80 people still online. That's amazing, because it's very, very difficult to sit through such a long time. I don't know how still people are sitting through. And one another interesting thing is that I've noticed that the person really utilized this, Professor R.V. Upadhyay, from from the very beginning, and he has noted down anything. If I have to talk about the whole talk, I just have to go there, because point by point, and I'm sure that some of the products is going to come from all these discussions sometime later. See, just a couple of points I will tell you. I'm not going to brief any of the talks what is given here, but if you have noticed one thing, all the talks are very basic research. That's what it has been given. Let me ask you a question. That do you know the number of patents that is being utilized in academic institution? It is less than 1%. It's somewhere 1 in 1,000 is utilized. So is the case with papers also. What has been mentioned is that, you can just see 1 in 10,000 papers in academics is translated. It's not because of that. When people start working on a project, they think that, eventually it can be they want to translate, or at least that is the concept. For example, structural biology, let's say identification of the protein or protein structure, 3-dimensional configuration, configuration. Why people are doing it's totally a basic research. Why people are working on that one is to identify the protein binding sites, whether it's a receptor binding, it's an enzyme, where is the ligand goes and bind. There's a lot to do, because if you are developing a molecule, to understand that 3-dimensional conformation is very, very important to generate a lead molecule. Without that, it is not possible. But developing that and finally identifying the 3-dimensional structure, then identifying a ligand and convert that into a molecule, eventually come into the commercial space is not that easy thing. Now, if you have seen that, the talks were concentrated on, let's say, nanotechnology, biotechnology, then agricultural foods, then structural biology by professor, a few structural biology talks were there. Then, a series of various basic talks were there. But then if you see, what we have given over here is a translational work. Now, what I wanted to tell here is that, a basic work and eventually getting into translator as a product is a very complex thing. Now, I was thinking, as I mentioned earlier, probably from the initial translation, discovery and developing is a complex thing. That is not. What I wanted to tell is that the last two part, especially something getting into the market. Now, I told about the patent. 99.9% patent has basically no value. One more thing I just want to tell, especially in academics and all, they start filing a patent very early. It has no meaning. You should never do that. Have you ever noticed that, how corporates or, pharmaceutical biotech company patents, they will not patent left and right just like that. When they find, for example, if you really see the patents, what we have filed, everything is translated. We will just not like that, never patent. When we are sure that, when we are going to translate this molecule, then we will go and file the patent. This is what all the pharmaceutical or biotech company. On the other hand, this is not what is happening in many of the academic institutions. Something is identified, immediately go and file the patent. It has no value because by the time it is translated, it will be, if it is a drug molecules, can be anywhere between 12 to 15 years. And the 15th year, the patent realistically has no value because only 20 years value is there. So never ever do that one. And the other thing, anyone who is interested in a academic research to be translated to a product and a commercial space, what you have to do is that what people want, what is the commercial space available. Even drugs, when you are developing, there are three different types are there. One is superior molecule, non-inferior, and inferior. If you are inferior, there is no chance. If it is something non-inferior, that means an existing product and to be introduced, you really will have to work through to introduce that in the market. That also is not easy. If something is superior, then you have all the chances, but then you have to go through a complex procedure. I just wanted to tell that, whoever is involved in basic research and, the identifications of mechanisms and everything, and if you are interested for translational research, product development, these things you will have to look into. I just wanted to tell that one instead of telling all those things. And thank you very, very much to everybody. And my sincere thanks to Professor Meenakshi Sundaram and all the people. They have been working very, very hard. And the first time I thought that, I have handed over this to, because last conference was done by Anirudh, which he meticulously, superbly done. This one is done by Priyanka as well as Kamalika, of course, support of many other people. Let me also tell you, see, we only have got 5-6 days to do that. It is not that easy to do in 5 or 6 days time to do, because we came to Professor Meenakshi Sundar about a week back. Then, suddenly decided, because the reason is that we wanted to launch the product. We have a series of products and we thought that, we should do it together and some kind of news. Basically, we need a lot of photos, other things, videos to put it into social media. That is the main reason. So we have all these things together. Thank you very much for helping us always, and to Professor R.V. Upadhyay. And let me also tell you that, we also have a PhD program, just in case, other students are there. I have a visiting professorship there, a professorship in their university and PhD guide. And few of our people, especially Priyanka, I know, is a PhD guide. And few other people are also there. We already have a very nice PhD program for translational research. In the translational research, it will be connected to a product development. Already somebody has joined. The advantage is that you have to pass UGC, CSR or that type of scholarship, which you can get anywhere between 32 to 37. Plus, we will give 25,000, which is as good as a very good job. Plus, every expense they are bearing. These are the things. So probably every year, 2-3 people will be taking, but it's not that easy to get, but somebody is interested, can contact. So thank you very much. And may I request Kamalika to give the vote of thanks. Thank you.
Good evening, all. On behalf of the organizing committee of Icon Bio 2023, I would like to take this opportunity to propose the vote of thanks. So, this seminar has been organized in a very short notice, as Ramchandra sir was suggesting. So, but all the people and guests and speakers, they have made it a very successful one. So at the outset, I thank our chief guest, Padma Bhushan Professor T. Ramaswamy. We are really enlightened with his knowledge and presence. We are thankful to our honorable guest of honor, Professor T.P. Singh, Professor R.B. Upadhyay, Professor Ajay Kapoor, and Sri Abhay Kumar Srishimal. Thanks to all of you for finding out time and from your busy schedule and gracing the occasion, sir. A very special thanks to our patron, Professor S. Meenakshi Sundaram. Sir is always there whenever we needed him. And Ramchand sir was suggesting that only maybe 10-15 days back we reached him, and as usual, he lended his helping hand to us. And we are immensely grateful to our chairman and conference patron, Mr. Sam Santosh, who never fails to encourage all this kind of initiative. And like any other events at our organization, Dr. C.N. Ramchand, CEO and CSO of MacGenome, he is there always. He is the driving force. And I would like to express my deep sense of gratitude to him also. We are sincerely thankful to all our eminent speakers who agreed to share their insightful research findings within a very short notice. A special thanks to the organizing committee and all the support staffs for their unflinching support and coordination. This event has been organized in a very short notice and also in hybrid mode that we have done for the first time. So I would especially like to mention the sincere contribution and efforts of conference convener, Dr. Priyanka, and my heartfelt thanks to all the members of the organizing committee, Dr. Shripriya, Dr. Runavo, Dr. Aniruddha, Swati, Suraj, Yadu, Joji, Ranga, Dakshina, Mahesh, Hampi, all the interns, Miss Arati from our Chennai and Kochi office. So last but not the least, I would like to thank all the offline and online attendees. I hope you all have learned something new and exciting, which will help you out in your future endeavors. I might have missed some people, some people, but yeah, definitely. Swati and Mahesh, you have done a wonderful job because this is the first venture we have done in the hybrid mode and excellent communication and troubleshooting. Thanks. Suraj is always there. Yeah, for all the thing, last minute presentation and all the things. And yes, all the comparing peoples also. So with this message, we move to the end of the today's seminar. We'll end the session with our national anthem. Let us all raise for that. No. We'll play from YouTube.
Your board is registered. Please, otherwise people will-
Okay. So before you part, we'd like to have all the registrants to come to our MacGenome office to have the dinner. Please come and that I won't tell. Okay, thank you. Huh? One line.
Online attendees, you thank them.
Yeah. Huh?